Hello, everybody. Happy Wednesday. I wish that I was here with you today, and I hope your day is going fabulous so far. So, our lesson today is a little bit of an extension on Punnett squares. We've had a few days of practice. I think you guys have been getting the hang of it. You'll have a little bit more extra time for practice today if you go through the notes. But this kind of Punnett square is called incomplete dominance. And I actually think the examples with this are almost a little bit easier than the genetics and the Punnett squares that we've been doing so far. Because as you'll see, instead of doing capital letters, lowercase letters, now we're only dealing with capital letters. So I think it actually is a little less confusing than what we've been doing. So in incomplete dominance, we have genes that are neither completely dominant or recessive. Okay, so on your note sheet, this is the very first definition that you have, right? It's basically the only definition that we'll be talking about today. Okay, so you're going to have these alleles that they're not really dominant and they're not recessive. So what happens is if both of them are present, they end up mixing together and forming a whole new trait, right? A whole new phenotype. Okay, so instead of like in pea plants where things were either yellow or green, with incomplete dominance, we actually do have a blending of the two. Okay, so they're always represented with capital letters. And as we go into our examples, you'll see what that looks like. Okay, so the only thing we're going to do for our notes today is we're just going to do examples. You already have your definition down, which was incomplete dominance. So now we're just going to do 3 examples together to show what this looks like. Okay, so the first one is in snapdragon flowers, which there's actually a pretty popular around here. You can kind of like move them so they look like mouths. I don't know if you know what I'm talking about. Anyways, one allele codes for red flowers, okay, so that's our capital R, and the other allele codes for white flowers, and we are using a capital W, okay? When a red flower plant is crossed, right, or if we were breeding the two, if we cross it with a white flowered plant, the result is a plant with pink flowers. So it is a mixture of the red and the white colors. Okay, and this type of genetics is actually way more common than the one we learned before, right? A lot of things are a blending of multiple. Okay? So for our example, if we're talking about the genotypes of the parents, okay, we have 2 types. So we would have RR, right? That's our red, because remember they still have 2 alleles. Okay, so they still have to have 2 always. And we'll have a WW. Okay, so we have a red flower and we have a white flower. So those are our phenotypes, right? We have red. Whoa. And we have white. Okay, so make sure that you are aware still of those differences between genotypes and phenotypes. Okay, this keeps coming back. So genotypes are the letters, phenotypes are what they look like. Okay, I'm sorry that my writing is kind of terrible. It's hard to do it on the computer. Okay, so if we're going to set up our Punnett here, 1 is on top, R, parent is on the side, W. Okay, so if we put them together, we have RW, RW, RW. And it doesn't matter if you have it RW or WR, it's the same. Okay, so our genotypes of the offspring in the boxes, the only genotype we have are RW. My phenotype is pink. Okay, for our second example, it gives us more of a question. It's kind of more of a word problem like we're getting used to. Okay, so it says, in some cats, the gene for tail length shows incomplete dominance. Cats with long tails are homozygous. Okay, so they have the same letter, they're capital L, capital L. Cats with no tails are homozygous, and they're represented by capital N, capital N. Okay, so cats with short tails are heterozygous. So it would be an L and an N. So it has one allele from each kind, right? Remember that we're mixing them together. Also, cats with no tails are definitely a thing. Your question is, what would be the expected genotypes and phenotypes in the offspring of a cross between two short tail cats? Okay, so both of our parents are short-tailed cats. So if we look back into our example, okay, cats with short tails are capital L, capital N. Okay, that's our two alleles. And we know the phenotypes are short because that's the question that we're trying to answer. Okay, so both parents, LN, LN. Okay, so we fill up Punnett square, so we'll have a capital L, capital L, capital L, capital N, capital L, capital N, and an N, and an N. Okay, so with this cross, we get every single possible genotype, right? So our genotypes of the offspring over on the side are what are the letter combinations that we have in our boxes. So we have a capital L, we have two capital L's, capital N's, and we have an N and an N. Okay, so we have a 25% chance that it'll have a long tail. We have a 50% chance that it will be short. And we have 25% chance that he will have no tail. Okay, our very last example for today is a little bit different. Okay, so our question is, what would the parents' genotypes be if 50% of the time their litters always had long tails? Okay, so this is a little bit of a higher level thinking question. Okay, so let's see what we know. Okay, what would the parents' genotypes, right, that's our letters, have to be if 50% of the time their litters always had long tails? So I actually would start with the Punnett square on this one. Okay, so we know that 50% is 2 boxes, okay? And our long tails are the genotype capital L, capital L. So I'm going to fill in 2 of my Punnett square boxes right away with those capital L's. Because in our question, it says 50% of the time we'll have long tails. Okay, so that takes care of it. Okay, so because of this, I know that one of my parents has to be capital L. Otherwise, there's no way I'm going to get 2 L's in each boxes. Okay, so then on this side, I also have to have a capital L. Because otherwise, we would not have an L here and an L here. It's not letting me mark in there, here and here. Okay, so the only other letter I can put with this one then would be an N. Okay, LN, LN. So our genotypes of the parents are LL, LN, means we have a long hair and a short hair. And the offspring, okay, we have an LL and an LN. So again, long, which is 50%, and short, 50%. Okay, that's the only way that it would work, where we would have 2 boxes that would give us the capital L, capital L's. Okay, if we were to cross it with a no-tailed cat, right? So if we had NN on the side instead of right here, then every single one of our boxes would be LN. So that would not work, right? That would be all 100% short. So we wouldn't be able to do it that way. Okay, so if you flip your paper over, you have some incomplete dominance practice. There's only 3 questions that you have to do. Okay, so it shouldn't take you too long. I want to get you started though on your key because I think there would be a little bit of confusion. Okay, so if we read it, it says Oompas, which is a fake thing, can have red, blue, or purple hair. The allele that controls this trait displays incomplete dominance, where purple hair is caused by the heterozygous genotype. Fill out the key for the genotypes and phenotypes. Okay, so we already have our phenotypes listed, red, blue, and purple. Okay, so our genotype for red would be capital R, capital R. Right, that would be for red. Blue is a completely different trait, so we're going to give that one capital B, capital B. Okay, and then our purple is the mix. Okay, so purple is going to be one of each. So we'd have an R and a B, which kind of makes sense because if you mix red and blue, it gets purple. Okay, so hopefully that helps clarify a little bit what different genotypes you should be using for the next 3 or 4 questions. Have a great rest of your day.